Hello, people. Welcome to developmental biology. I gotta tell you, right before I opened this up, I went over and I go to my son. I'll give you $100 to do this lecture. The notes are there, the slides are there. Just read the notes, show the slides. Wouldn't do it. Wouldn't do it. I went to $200. You still wouldn't do it. Went to my daughter, $50, and she went. She probably would have done it. Can you confirm the date of the last test? Well, let us discuss this, people. We could have the last test on the last day of class, which would be a Friday, 2 weeks from today, or we could do it during spring finals week. If it's finals week, I don't know the date. I haven't looked it up yet. I can look it up right now if you want. Just give me a thumbs up, thumbs down. Should I do a poll on whether we should have the exam early or not? Poll. This is the poll. This isn't yes or no. This is should we have a poll. Maria gives a thumbs up. Yes, yes. Thumbs up, thumbs up. Yes, yes, yes. Okay. I'll put a poll up in Moodle and we can decide whether we want it on that last Friday or on finals week. I have 2 exams the same date during finals week. Yeah, but Esha, you're so damn smart. You're going to ace them both anyway. So I don't feel sorry. Okay, I don't feel sorry. Esha Gupta never got less than a 98 in an exam. She's worried. Yada. Okay, so I do have a quiz for you today. A little bit, repetitive, but a quiz. I will open it up as soon as I'm through yakking here. I've got really one thing I want to talk about today, which is differentiation of muscle cells. And also as part of that, the difference between differentiation and proliferation on a cellular level. Okay, so, another thing. Apologize. I lost Wednesday's recording for some reason. I don't know why. I can't find it anywhere. I thought I had uploaded it already, but I can't find it. I'll keep looking, but I wouldn't expect much. Okay, so this is where we left off last time, which is how it is that once a cell starts down once a cell becomes determined to go in a certain direction, even though it's still dividing, all its progeny end up going down that same pathway as well. And it's partly because of the ability of genes to support themselves. So a cell, for example, in the somite gets exposed to a certain concentration of BMP and Wnt or whatever, and that turns on MyoD. MyoD comes up, and then MyoD causes transcription of MyoD. So you've got this positive feedback loop going that keeps MyoD on. And then once that cell divides, the daughter cells end up with some MyoD, and then that turns MyoD on in the daughter cells. So that kind of history keeps that MyoD going. And that those cells will always become muscle cells after this. And they become muscle cells as soon as you withdraw growth factors. So that's what we're going to talk about today. Why is that? Okay. So differentiation of muscle cells. So a little bit of review here first. So if you remember, and even if you don't remember, here's your somite, and we've got hedgehog coming up this way, and we've got Wnt coming down this way, and BMP4 coming this way. And that causes the somite to the cells of the somite to differentiate into different types of things based on what concentration of which hormone they're being exposed to. So this is the sclerotome. Sclerotome down here, that's going to be muscle. This is dermal myotome, which is going to become skin and muscle. So the part in the middle becomes the dermis. It's right under the epidermis, so that will become dermis or skin. And then you've got medial myotome and lateral myotome, and they will both become muscle. The medial myotome will become back muscles, and the lateral myotome then will move around the body and become kind of body case, I guess you would call it, or the muscles of the rest of the body. So this is what we're talking about, these guys here. So what happens? Okay, so you've got growth factors going around there, the growth factors we just talked about, Wnt and BMP4, and that's causing these cells to be undifferentiated, but they are determined to become myoblasts. And once those growth factors go away, and you can do this in vitro as well, they've been determined to be muscle cells. So as soon as you get rid of those growth factors, they become polarized, okay, which means narrow in the middle and long. Okay, so you've got different sides to them. And then they start to fuse with each other and become myotubes. And then eventually all those myotubes fuse together and they form one long muscle cell or a muscle fiber, right? A muscle fiber is a single cell. From origin to insertion, it's just one big cell. And it's a multinucleated cell because all these nuclei from the myoblasts remain, and you end up with one big nucleated cell. Will the quiz today cover today's lecture? No, it will not. But that doesn't mean go away. Stay with me. Evans, you got nothing better to do. Okay, so, oops, come on. Okay, so that is, so myoblasts then, the term blast means like a dividing cell, basically, an undifferentiated dividing cell. These are myoblasts because they're determined. They haven't differentiated yet. So what makes these myoblasts and not dermoblasts or whatever else kind of blasts? That has to do with several transcription factors. These transcription factors include MyoD. I think we've talked about MyoD before, but MyoD is a really important one. And then we also have MYF5 and MYF4. So these transcription factors are turned on by these, which we did talk about before. So PAX3 is a transcription factor that specifies somite in general, and then PAX3 and PAX7 start to make that somite into muscle. So these are going to kind of determine, do a muscle determination by causing these to upregulate. Now these MyoD and the MyFs are self-perpetuating, okay? So these are our myoblasts are still dividing, but we have that same mechanism I just talked about, keeping these transcription factors active in those cells, or at least being produced, constantly being produced. Then when growth factors stop, boom, you go into this differentiation pathway, which is controlled by this guy, MyoG, or myogenin, right? The genesis of muscle, myogenin. So, and also these are still up, and they're important for this differentiation as well. But these are going to cause the upregulation of myogenin, which is necessary. So this is kind of an important term in developmental biology. These transcription factors here, MyoD and the MyFs, are necessary, but they're not sufficient. So these alone, you can't get muscle without these, but these alone don't make muscle. Once you have these upregulated, they will upregulate myogenin or MyoG, and then that will cause, and MyoG directly makes muscle-specific proteins, things like myosin 2, which is the myosin in thick filaments, skeletal muscle actin, tropomyosin, the troponin complex, all the structural proteins you need to make a muscle will come up with MyoG. It's kind of like a master gene for muscle. And MyoG is only going to show up in non-dividing cells. Okay, it's only going to show up when the cell is differentiating. So basically, the MyoD and the MFs. Hang on, I've misspelled them in the notes. I have MRF and it should be MYF. So I'm going to have to fix that and upregulate the upload the notes again. But anyway, So the MyoD, MyoFs, and the MyFs are determining that cell to become muscle. But as we'll see in a moment, they're kept at a really low level when the cell is still in that blast state. And then when the proliferation growth factors go away, it goes down the differentiation pathway and upregulates MyoG to do that. Okay, but why is that? This is the question that's raging in all your minds right now. Why can you only have a cell dividing or differentiating? The cell can't go both ways. It can't keep dividing while it's differentiating. And that has to do with the cell cycle and cell cycle control. So we didn't do cell cycle in this class. You know, it's funny because it seems like all my classes are talking about the same stuff in different ways. So in cell biology, we talked about cell cycle and signal transduction, and we're doing that right now in cell biology. And then next fall in cancer biology, we'll be talking again about cell cycle and differentiation and non-differentiation, or de-differentiation, backing up on the differentiation pathway. And MyoD comes up again there. So it's all kind of mixed together. So I just want to give you a little bit of cell cycle review, which hopefully you got in Bio 101. If not, then you've had cell biology with me. And if not, you're kind of screwed. No, I'm just kidding. I'm not going to overdo cell cycle here. But you need to know about cyclins and CDKs. So we did talk about FGF receptor, which is a growth factor receptor. FGR, the hell, FGF, and then the FGF receptor. Okay, so we talked about this pathway already. FGF binds to the FGF binds to the FGF receptor, and they are tyrosine kinases, and they clump together and they cross-phosphorylate each other. And that produces these phosphorylation sites that attract the protein GRB2 SOS. So GRB2 SOS comes up, SOS then activates RAS. RAS then binds to RAF and activates it. RAF is a kinase and it activates MEK, or MAP kinase. MEK then activates MAP kinase, and MAP kinase goes into the nucleus. So that's the whole FGF pathway up as far as MAP kinase. Once MAP kinase goes into the nucleus, it's going to activate some transcription factors. One is called ETS, and the other is Jun. So Jun then binds to FOS, and these together, Jun and FOS, we call AP1. And this then will cause the upregulation of immediate early genes, which include MYC, Jun, and FOS. And those genes then turn on the delayed early genes, which include cyclins and CDKs. So for this class, you don't need to know all of this, but what you need to know is FGF causes the activation of MAP kinase, which downstream causes cyclins and CDKs to come up. Now, the CDKs are kinases. So the CDKs are going to actually phosphorylate proteins that actually cause it to go into the cell cycle. We'll see in a second. The cyclins then are proteins that control the CDKs. And the cyclins during the cell cycle will get broken down. The CDKs will stick around, but the cyclins get broken down. And then when they're needed again, they come back up again. So if the cell's constantly in the cell cycle, this mitotic cyclin here, or not mitotic, this would be the restriction point cyclin would help the cell to get past this restriction point and into S phase and then be degraded. And then the CDK is just kind of inactive until we get back around to this point again. And then the G1 cyclin comes back in again and activates it. So they're called cyclins because their concentration goes up and down during the cycle. So anyway, this cyclin CDK complex is going to phosphorylate a protein called RB. Now, E2F is the key to all of this because E2F is the protein that's going to activate gene transcription to get you into S phase there. All the enzymes you need to cause replication of the DNA. And once you start that process, the cell's committed to complete it and become 2 cells because it can't survive with all that extra DNA around. So when you get cyclin CDK active, it's going to phosphorylate RB, and RB is an inhibitory protein, and so it's blocking E2F. When RB gets phosphorylated, E2F gets released, and then you go into S phase. So that's what this transition is all about. The cell will be blocked here until you get enough cyclin CDK active that it will phosphorylate RB and allow the cell to pass that point. So that's cell cycle. That's in a nutshell. Basically, growth factor regulated cell cycle. You activate the receptor tyrosine kinases, blah, 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 MAP kinase, blah, 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 cyclin CDK, phosphorylate RB, release E2, and off you go into the cell cycle. Now, so this is constant in proliferating cells, right? Your cyclin's going up and down, but your CDK is always there, only active when the cyclin's around, blah, blah, blah. But at the same time, if we're talking about myoblasts, these myoblasts have already been induced to make MyoD and MyoF5, okay? And these proteins are perpetuating their own transcription. So they're there now, and they would cause differentiation, but they can't when the cell is proliferating because these cyclins and CDKs, the CDK is going to phosphorylate MyoD and MyF5. Okay, so as long as you've got an active CDK there, you're going to phosphorylate these. And then that targets them to become ubiquitinated, which we haven't talked about in this class either, but basically it just means it's going to target those proteins to be degraded. And so they're going to stay at a low level. They're always going to be perpetuating themselves, so they're constantly being made, but they're kept at a low level because you've got CDK there causing it to be broken down on a regular basis. Kind of like beta-catenin in that Wnt pathway, where it's constantly being phosphorylated by GSK3 beta, ubiquitinated and degraded. So it's always there, but at a low level. So that's myoblast. Okay, you're only going to be proliferating and not differentiating in the myoblast form. And so that's this bad boy here. So your cyclin CDK then is phosphorylating MyoD, which is a basic helix loop helix protein, dimer, gets phosphorylated and degraded. Now, what happens when you go into differentiation? Well, differentiation only happens when the growth factors go away. As long as you've got growth factors, you're upregulating this and you're breaking that down. When the growth factor goes away, oops, I missed something here. Oh no, that's what I wanted. Okay, so when the growth factor goes away, you no longer have this cyclin CDK complex. And so you're no longer phosphorylating MyoD, so MyoD starts to build up in the cell pretty quickly. Okay, when you're no longer breaking it down, it builds up. And it's going to then upregulate MyoG and cause differentiation. And just to ensure that we're going into differentiation, we're not going to divide anymore. Another thing that MyoD does, and MyoG does also, is it causes the transcription and then translation of another protein, P21. P21 is a CDK inhibitor. So it's going to block the cell at every stage of the cell cycle. So this is going to come over, block that CDK, and now the cell couldn't go into cell cycle even if it wanted to, like cells want things. But I couldn't go in the cell cycle anymore because it's being blocked by P21. So that's my story, and I'm sticking with it. Okay, let me just pull up my notes here real quick. So this thing's bugging me. Sorry. Okay, so bottom line is your MyoD transcription factors, MyoD, MyF4, MyF5, and myogenin. These are all necessary to become muscle. These first three are the ones that are going to be determination factors. They come up early because of hormonal influences, and once they come up, that cell is going to become a muscle cell, whether it likes it or not. There's some redundancy here. You can knock out MYF4 or MYF5 or MyoD, and you still get the same result. But if you knock out two of them, now you got some problems. It's not going to work so well. Typically, that embryo won't develop. So there's a little bit of redundancy with these transcription factors. And they're going to be expressed in myoblasts, which are undifferentiated and dividing. But they will upregulate myog or MyoG when allowed. And MyoG is found only in non-dividing cells, causes the transcription of your muscle-specific proteins, muscle actin, myosin 2, tropomyosin, troponin, blah, blah, blah. I'm doing a lot of blah, blah, blahing today. It's been a long semester. And this results in final differentiation. Okay, why does cell division of myoblasts block differentiation and vice versa? They are mutually exclusive. Either it's going to proliferate or differentiate. MyoD and MyoF are expressed in proliferating cells. They're necessary to get muscle, but they're not sufficient. Them alone are not going to cause it. These transcription factors are phosphorylated by CDKs, causing them to be ubiquitinated and degraded. And that makes it linked to the cell cycle. If the cell's in the cell cycle, MyoD and MyF5 are at a low level. Lack of growth factors causes the CDKs to downregulate, and then you get MyoD and MyoF accumulating, and they cause the production of MyoG, and then you differentiate. And they also cause the production of P21. And they also cause the production of P21 that's going to block any CDK that might happen to be randomly activated at this point. Not that anything's random. Cyclins and CDKs are no longer made when you withdraw that growth factor, so these build up, cause a transcription of myog. I shouldn't call it myog. MyoG turns on transcription of muscle specifics, okay? So that, unfortunately, is about all I have for today. I could have gone on to do, probably should have gone on to do blood cell differentiation, but we'll do that Monday. Okay, let's stop this share. Let me see your faces real quick. Faces. Alex is on the run today. Looks like you're on a bicycle.
It's nice out.
It's a little chilly over here on this side of the lake, but it is beautiful. Michelle Simmons doesn't care how beautiful it is out. She's got work to do. She's practicing her volleyball. No, she's not. Kwame never likes to let his face come up. That's okay. Okay, anybody else I can tease? Yusuf, can I tease you? Okay, I guess that's about all I have. I will hang up here and then I will, no teasing. That's all I do is tease. I only tease the people I like though. All right, so I'm going to close this out and then I'm going to turn on the quiz. You got 5 minutes for the quiz. One of the questions you've seen already, so it shouldn't be that hard. So it's a little bit of a repeat. So, all right, see you later.
Hey, Dr. Slish,
Yes.
when you get a chance, look at the email I sent you.
Okay, I'll check it out. See you Monday, the rest of yous.